Hi everyone, this is a mitosis lecture, trying it online first, and then you'll come into class and we're going to actually do some activities that reinforce what we've been talking about. Alright, so today what I'd like to do is talk about cell division. And in cell division, there's a whole bunch of issues that come with cell division, like stem cells and cloning. So let's go ahead and begin. Okay, let's talk about cloning real quick. Okay, so I have on the Haiku learning site a whole thing about the New York Times and a review about Dolly the cloning. Here's it in a nutshell. We basically took a cell from the udder, from a female sheep, and we took the nucleus, and we're going to place it into a egg cell. So the egg cell came from a different female sheep where we remove the nucleus from that egg cell and place the donor nucleus inside. We then hit it with an electrical shock. The cells began to divide and became an embryo. We then implanted that embryo into a third sheep who gave birth to Dolly. Now, she is an identical clone of the donor cell sheep. Now, this was brand spanking new research that had come and literally made a mammal, cloned mammal, and that's what made this quite unique. Now, a lot of things came from this. A lot of people said, oh, we can't have cloning of humans. If you ask the researchers at the time, they just wanted to make better sheep and better cows. So instead of doing the old selective breeding in the long term of trying to find the best cow or the best sheep, it would have been you have the best sheep and you would use this process to clone. Okay, so how do you end up with a sheep who is genetically different from the egg cell? And that process turns out to be cell development, or something we call differentiation. And here's what happens. So you have the first egg cell. It then divides into two. These two cells here are identical. Then those cells divide and they're identical. And then those 4 to 8, and those 8 to 16, and to 32, and to 64, etc., etc., etc. And it gets smaller and smaller, but the overall cell stays the same. And this process of development is quite unique because it turns out that these two cells are identical in terms of their DNA and their genetics, but they're slightly different. And when I get from here to here, each cell is even more differentiated. That is, if I just cut this in half and split it, I get twins. But if I were to divide these 4 cells into 4 individual ones, they would not develop into 4 individuals. In fact, none of these would grow into an individual. So this process is called differentiation. It's where each cell becomes specialized every time the cell divides. So in this case, here, undifferentiated, slightly differentiated, and then each step, each of these cells become more and more specific, each having their own job of what they will eventually become. So this differentiation is important. Okay, so let's talk about twins real quick, because it turns out there are 2 types of twins. Ones in which they share the same placenta, and one in which they have 2 placentas. These are called identical twins, and these are called fraternal twins. Now, what happens if these cells right here do not divide equally? And then you get twins in the news, or what we call conjoined twins. And so here, the 2 eggs never really separated, and you end up with conjoined twins. And then, of course, there's some fun.
I don't know if it's a boy or a girl yet because I don't know if it's a boy or a girl.
Face, another face.
breathing out both noses.
It is breathing out both noses.
Okay, so that completes our first lesson. And then what I would like you to do is go ahead and answer the 3 or 4 questions I have underneath, and then go on to the next set of videos.